cursor right until it appears here somewhere.
That's the second double.
Yeah, it's the second screen on the right.
Okay.
And then
Okay.
you go all the way here, hit alright, you know what to do.
Okay. Hello everyone. Today I'd like to talk to you about my project investigating the unknown functions in the minimal genome. So in 2016, a research group from Craig Venter Institute published a paper. A research group from Craig Venter Institute published a paper, Design and Synthesis of a Minimal Bacterium Genome, which presented the successful result of years of work spent on creating a cell that will contain only genes essential for life. To achieve this, they took a cell of Mycoplasma microbes, subspecies Capri, and using, it's not working. Okay, I need to be closer. And using global transposon mutagenesis, they identified genes that are not essential or genes that are not contributing to the robust growth. And then they deleted them, and then in a nutrient-rich medium, they grew a cell that has a genome of a size of 530,000 base pairs, 473 genes. And it's a cell that replicates autonomously and it duplicates in a reasonable time. So just to compare how small this amount is, the original Mycoplasma had a genome of a size over 1 million base pairs and 900 genes. And human genome has a genome size of over 3 billion base pairs and around 20,000 genes, or what we learned from yesterday's keynote talk. It depends who you ask. So the immediate question that was asked was, what are the functions of those genes that are essential for life? And to answer that question, they used genome context data, threading to known protein structures, and Tiger from families of equivalogs, and they tried to identify their functions. So as a result of that, they revealed that 149 genes, which represents 1/3 of the minimal genome, have unknown function. Each gene was annotated with a function which represents molecular function in gene ontology terms nomenclature. Also functional category, which is represents a biological process, and also they split the genes into confidence classes, representing how confident they were in the predicted function. So in the functional categories, we have, for example, the most numerous one, translations, which is a part of largest groups of genes responsible for expression of genetic information. We have also genes responsible for preserving genetic information, like DNA replication, also cytosolic metabolism, like proteolysis, and genes related to cell membrane, like transport, for example. And 59 genes didn't have assigned functional category, and they're all in unclear category. In the functional classes, we have equivalogs, probable, and putative. These three groups are genes whose specific functions were known with different confidence, with equivalogs being the most confident one, and putative the least confident one. Generic functional class is a group where we know a general function, but without, for example, specific substrate. And the unknown is they were not even confident about the general function. So these two groups, these are the 149 genes that we consider of unknown function. So the aims of this project was first to characterize the protein properties in these groups, to identify the differences, to find out why some of the genes, they were able to annotate some of the genes and some not. Then to use state-of-the-art computational methods to predict the functions of those proteins and identify the extent to which computational methods can be used to do this. So we started with identifying the differences between the groups. And to do this, we analyzed protein orthologs, domain architecture, transmembrane helices, disordered regions, and protein structural models. So we started with orthologs. Here you have a heat map of number of orthologs in all 5 groups across the 3 kingdoms, archaea, bacteria, and eukaryotes. So we can see that equivalent group is the most enriched in orthologs across all 3 kingdoms. That contrasts to unknown group, which has very few orthologs in eukaryotes or archaea, and a bit more in bacteria, especially in Therabacteria, to which Mycoplasma mycoides belongs to. So that suggests that the proteins might be clade-specific. Then we looked at domain architecture, and we have proven statistically that while with decreasing confidence of the prediction, also domain architecture changes. So this is exemplified the best by equivalogs group, which in all the proteins in this group have at least one domain, and almost 50% of them have multi-domain architecture, while in the unknown group, 75%, which is represented in this yellow color, more than 75% of them had no matches to known domains at all. We also looked at protein disorder, and here the differences did not reach statistical significance. However, the unknown group seems to be more disordered than the rest. Disorder here is represented, more than 10% disordered proteins are represented in shades of red. Then we looked at structural models. We predicted models using FIRE2. And while confidence templates were identified for all groups except the unknown one, the situation looks worse when we look at the protein part that could be modeled confidently. So here for only 9 of the 9 proteins, more than 75% of the protein could be modeled with at least 90% confidence. This is there is a huge difference between this unknown group comparing to the others. In the end, we looked at transmembrane helices, and here proteins that have at least one transmembrane helix are marked in brown, and generic and unknown group are enriched in those proteins that have transmembrane helices. Moreover, in the generic group, 24 proteins have at least 6 transmembrane helices. So to sum up, the proteins of unknown functions are depleted in orthologs and also known protein domains. They are enriched in disordered transmembrane helices, and they lack structural templates to model the protein structure. So with that information, we moved on to the second part of the project, to predict their functions and see if the computational methods can be used to do that. So I used various methods. We started with classifying the proteins into the different families, like TIGR-FAM families of equivalogs, also P-FAM families of domains, groups of orthologs using EGNOT mapper, functional families from CathDB. We also predicted gene 3D domain and structure models if possible, and where possible, with the basic idea of the function, we used methods that predicted gene ontology terms based on review of the second edition of CAFA. And then we moved on to membrane-related features and predicted binding ligands. So in the end, all the results were combined to predict the functions. And as a result of this analysis, we obtained confident predictions for 111 of 149 of these proteins with unknown functions. We were able to assign functional category to 29 of the proteins from initial unclear category, including 22 new likely transporters, 2 translation proteins, 2 cell division proteins, 1 from proteolysis, regulation, and chromosome segregation categories. And the final results suggest that more than 50% of the genome is responsible for preserving and expressing genomic information. And the second largest group is also cell membrane related proteins, which is around 22% of the genome. We were also able to move some of the proteins to higher confidence classes. 24, so here you have the colors represents initial functional classes and the bars current functional classes. So 24 of initial unknown proteins were moved to generic and 3 to putative categories. And 40 generic proteins were moved to putative categories. We weren't able to increase confidence besides putative category. So the 22 new likely transporters include 5 that are from specific type of transports, 2 ABC transporters, 1 ATP binding, 1 permease protein, 1 S component of ECF transporters, 2 that are likely to be from major facilitator superfamily, and 17 that are transmembrane proteins, a general transport. We didn't assign any specific type. So in the end, we looked at the confidence of results obtained from the methods to see what was the difference between the proteins for which we were able to increase confidence class and for which we weren't able to do that. So the colored squares represent the confident results and non-colored squares non-confident results. So here are the proteins that were moved from generic to putative confidence class, and you can see that for almost all of them we obtained results from almost all of the methods. Comparing to, for example, the unknown class, which remain in the unknown class, for which only membrane-related proteins mostly, except FFPRET and log3, which are non-homology-based predictors, returned results for those proteins. So in the end, I would like to show you the example of how predicting function looks like. So this is initially annotated membrane protein from unclear functional category in generic confidence class. And we suggest that that is likely to be a cation transporter from major facilitator superfamily. And we assign transport to its functional category. So TM-HMM predicted 13 transmembrane helices. FIRE2 modeled the structure based on a multi-drug transporter MDFA, which is a part of major facilitator superfamily. Gene3D predicted matching also to a major facilitator superfamily domain. And gene ontology terms, the highest terms that were predicted include cation transmembrane transporter, transmembrane transporter, or plasma membrane protein. So to summarize, we characterized the properties of those proteins with a known function and their lack of orthologs, domains, structure models, their enriching disorder and transmembrane domains. And even with sometimes not a lot of information, we were able to infer confident functions for 111 of them. However, I would like to emphasize that we still need methods that are not dependent on homology to tackle sometimes difficult cases of the proteins. like to thank Marcos and Martin Michaelis for work and supervision of this project, and the Genetics Society and National Science Foundation for funding me to come here. Thank you.
Any questions? Yes, Alex?
So thanks for a nice talk. The characteristics you looked at, did you consider looking at the rate of evolution of the different classes, or to put it another way, the level of conservation? Because that would seem to be an important correlate of whether you could find a function.
We didn't do that, but that's a good idea.
Okay, so one there, and then...
So what you're doing is you're making an assumption that the isolated function of each one of these proteins, if you will add up all those functions, will result in a living organism. But the
Yes.
possibilities that these entities, when you put them together, acquire some other functions. And so my question is, how could you deal with that possibility? That the life that you see is not a consequence of adding these activities.
So
Because if you think in this other way, probably the functions that you can predict are different than the ones that you are predicting based on comparison, sequence homology.
okay. Yeah, so I think I understand what you mean is that we obviously, we're not considering them together using those methods, and also the functions that we have in the minimal genome are context dependent, so they depend on the host, which is in this case medium rich of nutrients. But I don't know if it's possible to consider that as a whole. I don't know if there are methods that are predicting functions of the entire genome, considering also the relations between them.
All right. Thank you very much.
Thank you.
Put this in the car.
Thank you.